Hello, and thank you for joining us today for mRNA is Never Naked, exploring RBP target space and therapeutic opportunities, brought to you by Biospace in collaboration with Anima Biotech. My name is Lori Ellis, head of the Insights Biospace team, and I will be your host today. Before we begin, I would like to remind you that there will be a Q&A at the end of the presentation. So if you have questions, we want to hear from you. To ask our presenters a question, simply type your question into the chat window and hit submit. We'll be answering as many questions as possible during the Q&A. Today's session is being recorded and will be available to view on demand. You will receive an email with a link to today's session in it. I would like to now introduce our speakers. Dr. Iris Alroy, co-founder and CSO of Anima Biotech. Dr. Christopher Brannan. Dr. Brannan is an assistant professor and CPRIT scholar in the Center for RNA Therapeutics and Houston Methodist Cancer Center. And with that, the floor is yours.
Hi, everyone. It is my pleasure to host Anima's third webinar in mRNA regulation. This time, we're going to discuss RNA binding protein and their importance in regulation of mRNA lifetime. Before we start speaking about RNA binding proteins, I want to remind all of us why it is important to regulate mRNA translation. And this has to do with the way proteins are translated, how quickly, and the fact that the environment itself will change the way the ribosome translates the protein at the end of the day, how they fold. So we need to have environmental cues that ensure inside cells that cells are sensitive to that and stop or start translation as it is required. Because translation is a very energy starved situation, we need to ensure that we're not wasting energy in the cells or ATP. And because of that, the cells need to tightly regulate which proteins are being produced, which mRNA is being translated, and at what time. It needs to be sensitive to the amount of amino acids and tRNAs and responsible for the proteins that are need to be translated to these situations. In addition to that, it is important to translate the mRNAs where the protein is required, or in some cases in mRNA hubs, where protein complexes are co-translated at the same time that they are required. In order for the cell to be able to do all of that, there are many proteins that are helping both in a global manner, but also in a highly specific manner. So once mRNA is transcribed, it is never naked. It is always covered with protein, and essentially any protein that binds an RNA can be called an RNA binding protein or an RBP. So even RNA polymerase, for the sake of argument, is an RNA binding protein. For this webinar, we will only discuss proteins that regulate mRNA lifetime, transport, and translation, and thus RNA polymerase are not a subject of this webinar. So what are those RNA binding proteins are doing? So they are involved in mRNA processing per se, which include 5' and 3' and processing, which means cap and polyadenylation respectively. There are global machinery that are required for these processes, but there's also specific proteins that only regulate processing of specific mRNAs of interest. So there is a general splicing machinery, but there's also proteins that are involved in alternative splicing, which occurs only in specific cells or in specific tissues. On the 3' end, we have polyadenylation that takes place. About 50% of our genes have more than one polyadenylation site. So even here we have a specificity occurring. And all of these proteins together, or some of them as a subset, are involved in exporting mRNAs from the nucleus to the cytoplasm, and other proteins in both environments are involved in protecting the mRNA from being digested by endonucleases. These proteins are also involved in both decay promotion or essentially stabilizing mRNA, and again, they are responding to different environmental cues by being post-translationally modified. Many of the RBPs are important in anchoring the mRNA to specific organelles or to microtubules or filaments, and this helps to translate the protein at a specific cellular location. This is known in neurons, where we want to have proteins translated at neurons end, while the mRNA is actually made in the nucleus. And last but not least is translation itself. Ribosomal proteins are also RNA binding proteins. They bind RNA in order to translate it. And here we have initiation factor, elongation factor, and translation termination factor, which work in a general way together with other accessory proteins that are important for this. There's another family of proteins that are involved in tRNA processing, modification, and amino acid transferases, which load tRNAs with amino acid. But again, this will not be a subject of this webinar. A few words about the family of RNA binding proteins. So they're anywhere between 1,200 to 6,000 proteins, depending on how you find them and how you identify an RNA binding protein. Traditionally, RNA binding proteins were a group of proteins that have a recognizable RNA binding domain. But later, there are more elaborate ways to identify RNA binding proteins, and Christopher, which will give the second half of this webinar, will discuss new and novel ways to identify RNA binding proteins. From the known RNA binding proteins, which are today, there's about 1,200 of these. There are about 60 different RNA binding domains, which are divided here in this pie chart into these different names. The largest family has a domain that is called an RRM, and the other domains are listed as you can see here. An important attribute is the fact that many RNA binding proteins have more than one RNA binding domain from different families, and these are important in order to ensure a specific binding to different cis elements in the RNA, and also to have a cooperativity of binding. Because of that, usually using a single RNA binding protein to target in one long RNA binding domain doesn't always translate to a biological activity. In addition to the RNA binding domains, there are other domains that are important in the RBPs again, and this will relate to the different families. If it is a helicase, for instance, it will have, in addition to the RNA binding domain, a helicase domain, which unwinds the RNA. If it's RNA modifying enzyme, it will have a modification activity, and so forth. Another attribute which is found in many RNA binding proteins is the fact that they have a disordered domain. This means that this domain does not have a clear structure to it. And this makes the analysis of these proteins very complicated. It is known that these disordered domains are important for the interaction with RNA and the interaction with other proteins. And many of these RNA binding proteins are working in a very large complexes, and they are interchangeable. RNA binding proteins are also highly post-translationally modified, and I will show you a few examples how this regulates their activity. And they are localized to different cellular compartments, nucleus, cytoplasm, mitochondria, and so forth, again, because they are involved in different localization and transport activities of mRNAs. These are just two examples from our own project showcasing how RNA binding protein are regulated spatially. So this is coming from one of our projects where we've identified compounds that changes the post-translation modification of this specific RNA binding protein, and this is a 2D analysis of this protein using Western blot. You can see that in control situation, the protein has one specific charge, and it is not modified under this condition, but when we treat the cells with compound for about half an hour, we start to see the compound becoming more acidic because it is being phosphorylated. And this modification is important for activity. So under control condition, the RNA binding protein allows the c-MYC mRNA to be translated, and we can detect MYC protein. But although there's mRNA for p53, the protein itself is not evident because it's not translated. When we treat with the compound, the RBP is modified, p53 is made, and c-MYC protein is not made. We know that this is mediated by this RNA binding protein, also by the fact that when we use siRNA to this RNA binding protein, you can see that the compound is unable to rescue p53 translation. So just by using the same RNA binding protein under unmodified condition, c-MYC translation is allowed, but p53 translation is inhibited. But when it is now modified, we see that the opposite occurs. So one RNA binding protein can differentiate between proliferation and apoptosis. Another example is coming from another one of our project where we found that a compound changes the localization of an important RNA binding protein, which is under a regular condition is found in the nucleus, but upon compound treatment, it moved to these very specific granules in the cytoplasm. When it is found in these granules, the c-MYC protein is not being made, and the c-MYC mRNA is bound by this RNA binding protein, and it's found in these RNA granules, which are in which the translation cannot occur. So two different RNA binding proteins. One, the activity is regulated by the localization, and in the other case, by post-translation modification. We use high content modalities in order to study RNA binding proteins activities, because it's not only the amount of the protein or the weight is modified, but it's always also its localization, and specifically spatial localization, which is important to activity, and in many cases tells us if there's a disease situation, if an RBP is not functional. We have developed a full platform that is using a large language model to collect all available data in mRNA biology and store it in a modality we call Explorer. This we use in order to study the protein that are regulated mRNA translation that are involved in a specific gene or disease format. And this offers us the ability to mine data and start a project when we have this prior knowledge, and I will explain in a second how we do it. We then use it to devise a screen, and we have a knowledge graph and LLM behind the scene that guides the activity of the project moving forward, using all the data that was accumulated before, as well as the experimental data that we use in order to move the project forward. I just want to show you an example of how this is used. So this is coming from our MYC project. And in here, the system essentially analyzes all the splice variants of MYC and give us a snapshot of all the RNA binding proteins that are known to bind or to regulate MYC in the 5' UTR and 3' UTR regions. And then this gives us a list of these RNA binding proteins. We then can generate a library of these specific RNA binding proteins that are involved in MYC. So they're usually a list of between 100 to 300 different variants, and we can do it for any gene. And then we can ask the question, which of these RNA binding proteins is involved in cancer or in any other disease we are interested in. And by using microscopy, we can very quickly ask the question, what is affected, the protein, the mRNA, or both? So in this example, I can show you that one RNA binding protein, in this case, reduced the protein expression in the cells without affecting the amount of the mRNA. You can see in control cells it's very similar. There may be a difference in localization of the mRNA, and this is noted by the system. The other RNA binding protein that the system has identified also reduced the protein level. However, we see that there's almost no mRNA evident in the cells, and these are DNA probes against the RNA for the same protein. So basically, using this high content modality, that we can very quickly identify RNA binding proteins of interest that are involved in a disease process, and also have the first glimpse of understanding of how this RNA binding protein regulate the translation or RNA levels of this specific mRNA. We can also use this modality in order to understand what a compound we've identified in a separate screen is doing, again, fairly quickly. So this is an example from one of our projects where we found a compound that reduced the level of the protein, and we noticed these granules forming in the cytoplasm. We went back to our software and we asked the question, what are the RNA binding proteins that are involved in relocalization of this specific mRNA in the cytoplasm? So typically these will be RNA binding proteins that bind to the 3' UTR of the gene. So now a shorter list of RNA binding protein is produced. We ran again a knockdown screen looking at the protein and the RNA levels, again, in the presence of the compound, and we're able to identify an RNA binding protein that gave us the same phenotype that the compound gave us. So this specific RNA binding protein, when knocked down, also caused the MYC mRNA to accumulate in these granules. So again, very quickly, we could identify the target for this compound. Because we are working with many different projects, and as I told you in the beginning, RNA binding proteins have a plethora of functionalities as well as localization in the cells, we've decided to try and train an AI system and get an image classification for a biological function. And I want to show you how this was done. So the first thing we've done is using about 2 billion images that we've collected during the decade of work at Anima. We classify these images into each one of the mRNA regulation levels that we work with at Anima and that are involved in mRNA lifetime, starting from transcription through export, transport, decay, splicing, and so forth. And for each of these images from our different projects, we gave the classification. This is transcription, this is splicing, this is how mRNA relocalization looks like, this is how mRNA translation or localized translation looks like, and essentially taught the system based on the image to give us a biological function. And this is how the system classified a few of the images that we gave it after the training. So basically we have here images coming from different what we call visualizers or antibodies or probes for RNA. And for instance, this RBP has this specific phenotype, which is nuclear speckles, and indeed the system lists it as potential splicing or RNA processing modifier. In this example, we have an RNA binding protein that looks like it binds to microfilaments or microtubuli. So again, this is probably an RNA binding protein that is involved in localization and scaffolding. And basically using these modalities, the system is able, based on the images that we give it, to give us a potential molecular mechanism, and then we can go from there and very quickly validate or disprove it, depending on, of course, the biological effect of that we see. We use it both in our mechanism of action elucidation, but also when we start a project and you want to identify what is wrong in a disease situation, we can use a list of markers or visualizers, get an idea of what is different between disease and health, and the system will tell us what is specifically wrong in the pathway that are involved in mRNA regulation. And this is coming from one such experiment where we compare the disease and healthy cell phenotype using 30 different visualizers that were derived from the knowledge graph that we accumulated in RNA regulation. And basically what the system has done is found 4 different phenotypes that were involved or derived from the images that it studied. So these are the 4 different phenotypes, and this is essentially how you can see it at the end of the day. So healthy cells mainly have one phenotype that is associated with health, and while disease phenotype have 3 different phenotypes that are a hallmark of the disease, which means that most of the cells of the whale show this phenotype. We can then choose one of these phenotypes or 2 and run a screen and try to identify compounds that move this composite phenotype to a healthy environment. Again, all of the visualizers are antibodies to RNA binding proteins or probes to mRNA. So again, using this, we are able to do one of 2 things. So once we run it and identify which RNA binding protein is involved based on the visualizers we identified, if the RNA binding protein is known and the functionality is well understood, there's a structure, we can think of doing a target-based analysis on that protein using computational chemistry. The benefits are due to the fact that this is a known protein and we can have a very well-defined functional assay or in vitro assay, which we can run in high throughput and then validate it in our cell-based assays. The cons here are the fact that it may be that this protein is modified in some way that is not recapitulated in the test tube, and we will miss some of the phenotype that are associated with it. The other way that we are using it is by using one of our high content assays, either measuring translation of the specific mRNAs that are regulated by this RBP following the mRNA itself, as we show here. So measure the alternative splicing. This is the alternative splice variant of a disease versus normal, and we can look for compounds that change this back to the normal splice variants, or we can follow the localization of the RNA binding protein from an aberrant localization to the normal healthy localization event. I want to show you very quickly one example of such a project where we measured the translation of the MYC oncogene. This is a translation event seen here as a threat signal in the cytoplasm, and we identified compounds that inhibited the translation of MYC. These compounds show a dose-dependent reduction in MYC expression, and as I showed you before, we always look and compare protein to mRNA, and we found that one of these compounds specifically relocalized the c-MYC mRNA to these granules. Using the knowledge graph and LLM, we got a list of RNA binding proteins that were involved, and by using siRNA, we identified a potential RNA binding protein. One of the things we wanted to ensure is that this regulation is c-MYC specific and is not a global effect on all mRNA in the cells. So for that, we use probes for the poly-A tail of the c-MYC of all mRNA in the cells. We have a positive control, which is tubercidin, which is causing RNA granule formation for all mRNA in the cells, and this is followed up by a stress granule marker FXR1. However, the compound we've identified only send to granules a very small subset of the RNAs in the cells. It doesn't hamper transcription at all. You see that there's mRNA in the nucleus, and the granules that are formed are very distinct from these very large granules that are due to the stress that tuber seeding is inducing. So the phenotype is very different, and this is something that images can give you, well, gels or RT-PCR cannot show us. Finally, we did a additional validation. We followed the mRNA, MYC protein, and the RNA binding protein itself. We can show that the cells that have these granules do not express the c-MYC protein at all, as you can see here by these arrowheads. And the mRNA, which is found in these granules, is co-localized with the RNA binding protein to the same location. This is 24 hours of treatment. When we do sort of a movie and we try to see what happens first, is the mRNA goes to the granules first, is this RNA binding protein move to these granules, or what is the kinetics of this reaction, we get a very nice mode of action here. So basically, in the beginning, this RNA binding protein is found in the nucleus, and the granules are starting to form. And due to that, the RNA binding protein is now moving from the nucleus to the cytoplasm. And at 8 hours, it starts to form these granules. And at 24 hours, we see that the RNA binding protein completely co-localized with a stress granule marker. Again, wherever these granules are formed, the c-MYC mRNA goes into these granules, as I showed you before, and then a c-MYC protein is not being made. So when we look at the final modality of what this compound is inducing, we know that the c-MYC mRNA is being translated in these tumor cells under normal condition. Once the compound is added, the RNA binding protein is transported to the cytoplasm, bind to the c-MYC mRNA, and send it to these RNA granules. The stress granules protein are now being recruited into these RNA RBP complex, and the mRNA in these granules is now stored with ribosomes, but translation is inhibited and the protein is not produced. Finally, I just want to end with this slide saying that using the known knowledge in the field in mRNA regulation, we were able to pull all the data from databases, from articles, patents, also analyzing all the cis-regulatory element of RNA, and store it in a knowledge graph-based database, which we can query. We query it in order to select visualizers or RNA regulators or probes, and based on that, train our AI system to identify what is the signature of disease, and based on that, devise different screening capabilities to identify compounds that can remedy the disease to healthy phenotype. In order to do that, we need to have good information about this whole RNA regulatory field, and one of these is to get sufficiently good information about what RNA bindings proteins are and how they are built. And Christopher, in his slide deck, will show you how his group was able to do it in a very elegant manner.
Okay, thank you, Iris, for your excellent talk and the kind introduction. And I'm honored to have been invited today to talk about my work at the Center for RNA Therapeutics at Houston Methodist Research Institute, which is focused on RNA interactions with RNA binding proteins, or RBPs as I'll refer to them, and RNA-based therapeutics. Okay, so today I'll give a broad overview of some of the work that motivated the current projects in my research program, which was launched a little over a year ago. And I'll describe one project that uses RBP-focused protein-protein interaction networks to discover new RBPs and new RBP functions. I'll then describe another project that uses RNA-protein interaction detection technologies to explore translation control mechanisms during cancer progression and treatment. And my group is now using insights gained from these studies to design and develop new RNA-based therapeutics that I won't have time to talk about today. So my work is broadly focused on RBP-RNA interactions that orchestrate the mRNA life cycle depicted in this diagram. So every step of this cycle is associated with some RBP mutation or dysfunction that results in human disease. RBP discovery efforts have estimated that nearly a quarter of the human protein coding genes code for RBPs, and over 1,000 of these RBPs are associated with human genetic disease, more even than DNA binding transcription factors. So there's a medical need to fully catalog and functionally characterize this class of proteins. The specific mRNA life cycle steps that my group is focused on are nucleocytoplasmic transport, or NCT, which is often disrupted in neurological disease, and translation by the ribosome and associated factors, which is often disrupted in cancer. So the story of how I became interested in nucleocytoplasmic transport, or NCT, factors began with this pilot study I conducted as a postdoc in Jean Yo's group in 2016. This study set out to discover and characterize RBP protein-protein interactions, or PPI networks, using a panel of 12 RBP baits for affinity purification, followed by mass spec analysis of interactions with and without RNase treatment. Within the resulting network, we observed that RNA-mediated interactions with RBP baits enriched for other RBPs. So we used this observation to construct a simple computational classifier, but predicted RNA binding activity for a protein of interest from any PPI network. The classifier worked by counting the RBP neighbors of a protein of interest at 3 levels to generate an RBP prediction score. This classifier term SONAR was the best performing classifier at the time and successfully predicted the majority of annotated RBPs within our network and the entire genome. Excitingly, it also predicted nearly 1,000 new RBPs that had not been previously annotated. One class of proteins within this large candidate list was the NCT factors that compose the nuclear pore complex and associated import and export factors. Other studies at the time had revealed that these NCT factors co-localize and co-aggregate with well-known RBPs under conditions of stress. We performed cross-linking and immunoprecipitation, or CLIP, on a number of these NCT factors to validate their RNA binding activity, and we observed specificity of binding to mRNA features, including coding sequences, untranslated regions, introns, and conserved specific sequence motifs. To expand our RBP-focused PPI network beyond the scope of the initial bait panel of HNRNPs, in collaboration with Marko Jovanovic's group at Columbia, we performed IP mass spec in the presence or absence of RNase to obtain the interactomes for over 100 RBPs, representing complexes across all steps of the mRNA life cycle. To do this, we utilized the large collection of IP-grade antibodies shown here, which represent well-known RBP complexes that function at each step. This expanded RBP PPI network contains 3 types of interactions shown here and denoted in this key. These interactions included direct protein-protein interactions that are RNase insensitive, RNA-mediated interactions that are removed upon RNase treatment, and RNA-shielded interactions only observed upon RNase treatment. Previously reported interactions are green edges, and we found the expected direct interactions for many RBP complexes, such as the exosome, and found expected RNA-mediated interactions with many RNP complexes, such as the spliceosome and ribosome. We augmented this network with an orthogonal approach called size exclusion chromatography, followed by mass spec, that allowed us to both validate our IP'd interactions and infer prey-prey interactions from complexes that co-elute from the same size fractions. Heat map of the correlation of all preys in the network produced dense clusters corresponding to well-defined protein complexes that function across the life cycle. And since many RBPs clustered across multiple life cycle steps, we calculated a betweenness centrality score, and RBPs with high betweenness centrality scores were predicted to be multifunctional RBPs that bridge RNA life cycle steps. And we were able to validate implied new functions for top scoring candidates by orthogonal approaches. So to update the computational portion of the classifier that uses these updated PPI networks to predict new RBPs, we first augmented the sonar portion of the classifier to train on a broader collection of PPI networks and to penalize proteins with low network connectivity. We then added a primary amino acid sequence analysis component referred to as HydraSeq, utilizing a k-mer-trained support vector machine and a full-length sequence-trained convolutional neural network. And we then combined and integrated all scores to arrive at a final Hydra RBP predictive score that outperforms both the original sonar classifier and all RBP classifiers to date. We next applied a widely used method in the computer vision field called occlusion analysis to the HydraSeq component of the classifier. By occluding a sliding window of 20 amino acids from the protein sequence and comparing the output score with a full-length protein score, we could calculate a delta score that reports the contribution of the occluded region to the protein's RNA binding activity. Occlusion map analysis robustly detected known RNA binding domains, as evidenced in these example RBPs, FOOS and RBFOX2. Excitingly, Hydra predicts nearly 1,500 new RBPs and 76 new RNA binding domains to be experimentally validated. So my group is now interested in using these updated RBP PPI networks and RBP classifier to prioritize and characterize the RNA binding activity for a curated list of roughly 60 NT-CT factors. Of these factors, 90% have been detected by RNA interactome capture, 98% are predicted by our Hydra classifier to be RBPs, and almost none have been evaluated for RNA binding specificity by CLIP or other methods. So we've collected IP-grade antibodies to perform CLIP for these candidates, and we're working our way down the list. One issue that we face is that the CLIP methodology, while it's powerful, is difficult to rapidly scale for many new candidate RBPs. So to prioritize candidates, we will use a scalable orthogonal approach termed STAMP. The STAMP strategy is to fuse an RNA editing enzyme to an RBP of interest and inducibly express this fusion in cells. The resulting RNA edits mark RBP target genes and RBP binding sites on those genes. Since STAMP uses standard RNA-seq, it is the first single-cell enabled approach for obtaining RBP binding profiles. This multi-RBP single-cell experiment shows RBP-specific cell clusters based on editing readout of RBP binding profiles in each cell. Using these single-cell editing readouts of RBP binding, we can extract the RBP-specific binding motifs even when downsampling to a single cell, which shows unprecedented specificity and sensitivity. We can also use long-read sequencing platforms to show isoform specificity of binding, as exemplified here for the two different 3'-METR isoforms of the FAR1 gene, where we see edits deposited by RBFOX2 stamp only on the long isoform, and this specificity would have been invisible to short-read methods such as CLIP. Stamp with ribosomal subunits allows measurement of translation and translation perturbations, as shown in these scatter plots that reveal that treatment with mTOR inhibitor drugs, which modulate translation but not transcription, results in reduction of ribostamp edits on drug-sensitive genes with no corresponding change in transcript levels. This ribostamp approach is also single-cell enabled and detects ribosome association on individual genes in individual cells, as shown in this browser track from a standard 10x single-cell experiment. And single-cell ribostamp is in good concordance with orthogonal bulk measurements of ribosome occupancy, such as polysome profiling. So the single-cell ribostamp approach was used in vivo to measure the translation landscapes in a xenograft model of breast cancer. An RBP-focused CRISPR screen identified RBPs that convert survival to make hyperactive cells, and upon depletion of the hit RBP YTHDF2 from this screen, ribostamp revealed the translation response from xenograft tumors at single-cell resolution. This screen identified a number of other translation control factors that can be individually evaluated for effects on cancer phenotypes using this single-cell ribostamp approach. We have since scaled our stamp approach by cloning these hit RBPs along with all the translation control factors shown here into our stamp lentiviral expression vector. This construct library includes all eukaryotic initiation factors and ribosomal subunits. And hundreds of stamp experiments can be done in 96-well format using this library of constructs. This heat map shows the average metagene transcriptome-wide binding profiles for all EIFs and ribosomal subunits, which showcases the scalability of stamp multiplexing. And this type of multiplexing can also be done in pooled single-cell experiments. So this UMAP visualization of single-cell multiplex stamp experiment shows that clustering by edits for ribostamp, control stamp, and a pool of many RBPs transduced at a low multiplicity of infection. So we're now working to apply this powerful single-cell multiplexed approach to obtain many RBP binding profiles in one-plot experiments in vivo. So single-cell analysis approach called RNA velocity allows us to infer cell state transitions from single-cell RNA-seq data. Our collaborator at Houston Methodist, Guangyu Wang, recently developed a scalable deep neural network that locally infers RNA velocity for individual cells from neighboring cells to relay a series of local velocities and infer velocity kinetics. So we're now working with him to add single-cell ribostamp to this RNA velocity framework and enable the first total translatome velocity. And we want to apply this combination of RNA splicing and translation velocity to measure cell state transitions during cancer progression and evolution of therapy resistance, which rely heavily on rapid proteome rewiring using translation control mechanisms. And because cancers hijack RBP function to modulate translation, there are many chemotherapies that target translation machinery. So we're working with our collaborators, Jenny Chang at Houston Methodist and Na Zhao at Baylor College of Medicine, who are inhibiting translation machinery in syngeneic and patient-derived xenograft models of triple-negative breast cancer. And in these models, we're measuring RBP-RNA interaction and translation responses to these treatments using single-cell stamp. In future work, we'll continue to combine RBP proteomics approaches with machine learning algorithms to discover and characterize new RBPs and RBP functions. And we'll update and utilize single-cell RNA protein interaction approaches to evaluate multiplexed RBP binding behavior for in vivo disease models. And the ultimate goal is to pinpoint the intersection of mRNA regulatory factors, NCT and translation machinery, and drivers of neurodegeneration and cancer to identify potential new therapeutic targets and modalities. So with that, I'd like to thank the Yo Lab at UCSD, where we did a lot of the work I showed today, and our collaborators at PacBio and Columbia University, and my group at Houston Methodist, our collaborators at Methodist and at Baylor, and our funding.
So I want to remind you that we're continuing our webinars around mRNA biology, and the next time we will showcase how we use our new platform of machine learning around different areas in data mining and image analysis in order to strengthen target identification as well as mechanism of action elucidation in this novel and emerging field. of therapeutics. Thank you.
Well, I want to thank you both for that informative presentation. It is now time for our Q&A. So I invite you both to turn on your cameras as a few questions have already come in. Okay, so for the first question, what is known about the binding of some of these proteins to non-coding RNA, even though it does not code for any protein?
So yeah, I'd be happy to try to answer that question. There's a lot known about RBP binding to non-coding RNAs, and it happens in the nucleus during transcription and processing of the long non-coding RNAs, by the normal transcription machinery. And then there are proteins involved in retaining long non-coding RNAs in the nucleus and in the cytoplasm, and even, preventing translation in the cytoplasm. So they're always interacting with protein. It's just a different set of proteins usually than those are translated, or that are protein coding, because those interact more frequently with ribosomes in the cytoplasm.
Dr. Alroy?
I don't have anything to add there. We are mainly working with coding mRNAs, so we have less experience with non-coding mRNA.
Okay. So then the next question from our attendees is for you. Regarding to the first part of the presentation, does the set of RNA binding proteins include splicing factors, and is the high content screening also applicable to these splicing factors and pre-mRNA splicing regulation?
So the answer is yes. At Anima, we're working with all RNA binding proteins, irrespective of their functional role. And we're following also splicing machinery, as well as proteins that are responsible for alternative splicing regulation of target genes. We also have designed assays and screen where we follow alternative splice variants, specifically in neurodegeneration. There's many cases like that, and we're checking for rescue of a missplicing event in these cases. So the answer is we can follow both the proteins and the splice variants,
Okay.
specifically in high content. And
Well, then I also have another question for you. Are you planning to incorporate new spatial transcriptomic approaches to your visualization workflow? And regarding your high content visualizations and perturbations, sorry for that mispronunciation, do you only rely on cell line models, or can you use patient-derived cells or even in vivo models?
So as far as spatial transcriptomics, this is still too complicated for us to achieve. We are doing analysis of several mRNAs by using fish probes, but we haven't done spatial transcriptomic yet. We are following the field, and we see that there's a lot evolving there, and enabling many different markers at the same time and analysis at the same time. So we probably started with a low throughput and work our way through this. As far as the cellular environment, so we have worked with patient-derived cells, both fibroblasts, iPSCs, neuron that are derived from these iPSCs. So any cell that can adhere to plastic, we can work with and are working with. So it's not specifically only cell lines.
Okay, the next question. What's the specificity of RBP interacting with RNA? Any RBP models only RNA of a single gene when perturbed? And I believe that's for either of you.
So Christopher, you want to start, or should I?
I'm not sure if I fully understand the wording of the second part of the question, but the specificity for RBPs for their target RNAs, it varies from RBP to RBP. Some have very high specificity, some have lower specificity, some have conserved sequence motifs that they bind and bind only specific features of the RNAs. So yeah, it's kind of RBP specific, and you can get that specificity by, sequencing the RNAs that you can immunopurify those RBPs. But the second part, maybe you can handle or try to interpret.
I can try. So just to add to your answer, we also see that although in some cases it seems that the sequences that are recognized by RBPs are pretty redundant and seem to bind or be found in many RNAs, there's actually a high specificity in many cases. This may be due to which RBPs are expressed in the cells. Many of them are work as a complex, and this confers additional specificity. They are also highly modified, as I showed in my presentation, and this modification will also influence the way they can bind to target RNA. So this is something that I think is still not completely understood and appreciated. If you knock down a specific RBP, you do not see a general disappearance of many RNAs in the cells. If you pull down, you do see, I would say, between tens and hundreds, depending on the RBP that you're interested in. So again, it will be depending on the RBP, but overall, we don't see thousands of mRNAs changing when you knock down a specific RBP. And again, as Christopher said, this will depend on the RBP of interest.
Okay. The next question is for Christopher. The Hydra method, which predicted RBP and RBP-PPI, is very impressive. Was it incorporated into public RBP or PPI databases?
Yeah, the Hydra method was published this last summer, so it's publicly available. The code is available, the data is available, and then the scores, the RBP and RBD predictive scores are available in table format. As far as I know, it hasn't been incorporated into any existing, I guess, what did they ask, pipelines or databases? But it's definitely available.
Okay. The next question is for you, Iris. Regarding your MYC specific stress granule forming compound, I guess since these granules have stress granule marker proteins like FXR1 in them, it would be surprising if they only had MYC mRNA in them. I'm guessing you eventually use some orthogonic global omics methods to determine the specific off-target effects from a compound like that.
Yeah, so this is related to our MIC project. So indeed, when first we saw that, we were worried that this is a general stress granule formation in response to a stressor in the cell, so maybe the compounds are stressors. And we really profiled it a lot, and we're surprised to see that these granules only hold MYC and additional 10 or 20 other mRNAs. We saw it by doing an RNA-seq and looking at the influence. And so this is specific to the RBP that actually is affected by the small molecule. And the FXR1 is being recruited to these granules only after 24 hours, and it's actually the RNA binding protein that is affected by the compound that is recruited first, and as times evolve, then these granules evolve. But we don't see a general effect on many mRNAs in the cells, but only really a subset of cancer-related oncogenes.
Okay. The next question we have is for Christopher. STAMP methods seems to overcome challenges in RBP immunoprecipitation methods, such as ICLIP, and mRNA protein interaction methods, mainly the requirement of good immunoprecipitating antibody and high background. However, the use of overexpression of a tagged RBP may result in improper localization or interactions with mRNAs, as it does not naturally interact with. Can you comment on the overlap in targets between endogenous and this overexpression system?
Sure. Yeah, that's a good question. So it's something that we think about when we do these experiments. Definitely, it's important to characterize your RBP editase fusion and how it localizes in cells using, IF and quantify its expression compared to endogenous. So those are all important things to consider when doing these experiments. The expression levels can be dialed, and so can the localization. So you could sort of more mimic the physiological localization and binding profiles. But we see pretty good target overlap. One of the main issues is that because, we do this with mRNA generally, mRNA purification kits, we tend to lose sort of intronic binding profiles. So we get good target overlap, but not always great peak overlap when doing nuclear splicing factors. But for ribosome, and cytoplasmic factors, the overlap for CLIP is usually quite good. And we're working out some new, I guess, parameters to try to better mimic nuclear binding profiles.
So the next question that I have is for Iris. How does the fact that different cells express different genes contribute to the selectivity of drugs that target RBP function? And what types of proteins in the cell regulate RBP function / selectivity, localization, and stability?
So this is also related to one of the questions I saw from the audience around the disorder domains in RBPs. So it's not completely clear, but it does seem that this disorder domain control the activity and localization of RBPs, and probably modification by phosphorylation, by methylation. So we do also see methylation, ubiquitination. All of these will regulate the localization, the complex that the RBP is found in, and because of that also the specificity. And as I said, because these RNA binding proteins have different domains, these complexes may give another layer of specificity, because RBP domains from these different proteins will bind target sequences, and then we will have some sort of like a Lego-like effect and the composition of a new sequence recognition. And the fact that these proteins are expressed in different cell types and tissues in different manners will give another layer of complexity here, which we can use when we identify inhibitors of these complexes.
Okay, I think we have time for one more quick question. And Christopher, I'm going to direct it to you. In the single cell STMAP methodology, what is the time between running the experiment and getting the final analysis? Is the output a long list of targets or a more specific one? And do you require network analyses?
Yeah, so the analysis pipeline for the single cell STAMP approach is essentially the same as would be for a standard single cell RNA-seq experiment, except that you have to quantify the mutations or the edits that are deposited. So that portion of the experiment takes a little bit more computational time. So, at the moment, I would say that adds a week or two to the computational time, but everything else in the pipeline remains the same. And so you do get cell by gene tables with the metric that you've defined for your editing. You can specify where that is measured on the RNA. So basically you can measure both expression and then the interactions using the same exact workflow.
Okay. Well, with that, it looks like we've run out of time. So I want to thank you both, Dr. Christopher Brannon and Dr. Iris Alroy, for that great presentation and our Q&A. On behalf of Biospace and our sponsor, Anima Biotech, we hope you have enjoyed this presentation and have a wonderful rest of the day.